doing this?
Yes.
Okay. All right. So let's go ahead and get started here. So
I'm going to give just a quick introduction, if I could. I understand time's limited, but I just want to highlight that this portion of the meeting is to provide a briefing on Florida Gulf Coast University's research on red tide presently affecting Southwest Florida region and the connection to our CCMP water quality improvement action plan. It's also tangentially related to the hydrological restoration through salinity, fish, wildlife, and habitat protection through aquatic habitat impacts and public engagement. And so with that, Dr. Parsons, I turn this presentation over to you. Thank you very much for joining us.
Great, thank you. All right, next slide, please. So basically, there are 3 programs that I'm going to talk about briefly today. Some of the monitoring efforts we're doing, looking at removing fish as a mitigation strategy, as well as looking if after red tides, if brevitoxins, the toxin produced by red tide, basically remains in the environment. How resilient is it, and will it be dispersed and distributed within the local food webs? Next slide, please. So based on our current knowledge with red tide, one of the things that's really confounding with red tide is that we think it initiates offshore. So you can see the red rectangle up in the upper right here. And about 50 miles offshore, we think we get an upwelling of deeper shelf waters that bring in nutrients that actually first stimulate red tide. As it then moves closer to shore, it needs to be synchronized with a Saharan dust event where dust blows across the Atlantic Ocean and across Florida, which brings in iron. This iron basically fertilizes the water and allows a cyanobacteria called trichodesmium. Trichodesmium produces, it's a nitrogen fixer, so it will actually produce organic nitrogen, and that organic nitrogen in turn continues to feed red tide. So the slide's frozen on my end. I don't know if you guys can still see it, but as red tide continues to move closer to shore, it uses this organic nitrogen, and then the recycled nitrogen multiplies even further. By this point, it's highly concentrated and can start killing fish. As the fish degrade, it produces even more nutrients, causing red tide to multiply even more. And now by this time, it's close to shore. And so here's where the big question is, what role does human activities and our water quality play in red tide? So the important thing to keep in mind here is, again, based on our current knowledge, it looks like red tide initiates offshore, a completely natural event. But as it moves inshore, what does water quality, what do our activities, how do they influence red tide? Do they make it worse? Do they make it longer? So on and so forth. So those are the real big questions that, as I guess, a research community, as well as a stakeholder community, that we're dealing with. Next slide, please. So this is just an example of some of the difficulties. This is a very simple graph. Basically, it's looking at annually averaged nitrogen loading at S79. That's on the x-axis, versus the number of samples, or the percentage of samples, where you basically have high concentrations of red tide, Karenia Brevis. And we're defining high concentrations as 100,000 cells per liter, which is about the concentration that you would expect to start seeing fish kills, for example, and experience respiratory irritation. And so basically what this graph is showing is that on an annual basis, when you have higher nitrogen loading events, you don't necessarily see higher percentages of Karenia Brevis events. And so it's not a simple linear relationship. And this is what really makes red tide difficult to study, because you would expect it to basically have a nice linear cause and effect if we were seeing this direct relationship with nutrients. But it's convoluted, it's complex, and that's what's made it very difficult to really link the two together. And that's what a lot of research has been going into for years, to try to really strengthen this relationship so we can better combat red tide and better predict how red tide will react to improving water quality, which is all of our goals. Next slide, please. So some of the things that FGCU has been involved with the establishment of the water school, the state and FGCU have been really funding environmental research at FGCU through the water school. And it's given us more capabilities. So for example, this is one of our new boats. It's a 31-foot twin V, and this boat allows us to go pretty far offshore. We routinely go out 75 miles offshore to collect samples for red tide. Next slide, please. So we were recently awarded a contract by the Fish and Wildlife Research Institute to establish a third monitoring transect line. So you can see their original transect line on the top here shown in red. This is called the Rome line, which is the routine offshore monitoring effort that is basically heading west out of St. Pete. You can see the line, the Rouge line or the Rogue line, the routine offshore Grangian effort that Moat runs on the blue line off of Sarasota. And now we have established this line for Southwest Florida, basically running from the Sanibel Lighthouse going 75 miles offshore. And if you drew a line east-west, this would basically be 60 miles offshore of Marco Island. So we haven't had this routine, this is a monthly sampling effort. We haven't had this routine monthly frequency of being able to sample like this offshore. And not only are we taking surface water samples, but we're taking a mid-depth and bottom, as well as if we see high concentrations of chlorophyll, because we know red tide will actually sit as a layer, almost like a layer of icing. I guess it would be toxic icing, no, at mid-water depth. So it's not always just at the surface or just at the bottom. It may be in a very thin layer somewhere in between. And that's what makes, that's another difficulty in terms of trying to track red tide. Next slide, please. So we put out sediment traps and we put out monitoring gear. We'll be putting divers in the water, and basically we're continuously collecting data electronically, and that will help us measure some of these deeper water movements, some of this cooler water that's moving in from offshore, to really continue to test this hypothesis of an offshore initiation zone. Because as all of us know, all of a sudden red tide basically just shows up. It goes from almost nothing to a bloom just like that. By the time we detect it, it's almost too late to really react to it. It's already blooming. And that's a real hurdle in trying to mitigate the impacts of red tide and trying to better understand what leads to that initiation. Next slide, please. One thing that's really interesting in this effort is on the left is a schematic that Karen Steidinger put forward about 20 years ago. Karenia Brevis, the current name of red tide, is actually named after Karen Steidinger. She put her whole research career to study Florida red tide, also known as Karenia Brevis. You can see with this figure, back then it was called Gymnodinium brevis, so they changed the taxonomy of this. I'd like you to take particular attention or pay particular attention to the bottom left where you see that 2N and that I with a question mark. That represents an unconfirmed presumed hypnozygote. That means a resting cyst or a spore. And so some of us really think that the reason why red tide all of a sudden shows up is because it has a resting stage in the sediments that all of a sudden germinates. And then those cells rise up out of the water column and boom, you have a bloom. The middle panel, you can see the two basically balls, if you will. The top ball is a picture of Karenia brevis forming a pellicle cyst. This is not a resting cyst. This is more of a stress-related, trying to survive life stage. The bottom one with the K on it, and you can see that little black triangle, that's a closely related species called Karenia micimoitoi, which is also present here in the Gulf of Mexico. But this is a true resting cyst. So other members in the genus Karenia can form a resting cyst. And we have taken pictures of cells that are at least a pellicle cyst, but maybe are approaching this resting cyst stage. On the right, you can see this picture is now probably 2 weeks old. And this is one of these balls, one of these pellicle cysts from Big Carlos Pass. And so we do see evidence of, you know, potentially reaching that I question mark stage. And so there are ongoing research efforts with FWRI and other groups to try to really further understand this and see if red tide actually forms cysts. The other neat thing about this is this might help explain why red tide all of a sudden disappears. Because if they form these cysts, they're just going to sediment out of the water column and bam, the bloom is gone. So the cysts would germinate, you get a bloom, the cysts would form, and you would end a bloom. So it would really help explain this, you know, start-stop action, but we just don't have enough data to see if this is truly a mechanism. That brings up the next question then, what causes the blooms to germinate and what causes the blooms to encyst or the cells to encyst? So there's a lot of work, as you can see, that needs to be done even on these life cycle stages. Next slide, please. So the second project I'd like to talk about a little bit is working with Cindy Heil up at Moat Marine Lab. And this is looking at what if we remove the fish carcasses while they're still floating in the water and, you know, basically releasing nutrients as they decay. What if we remove them from the water and remove that nutrient source? Would that actually lessen red tide? Next slide, please. So the whole premise is when you get the blooms forming, when they start reaching about 100,000 cells per liter, they start releasing enough toxin into the water that will start killing the fish. As the fish then start decaying, they'll start to release nutrients. These nutrients can then feed back into the red tide, which will cause them to multiply even more, producing more toxin, killing more fish. So you get this vicious circle. It's going to be a positive feedback loop. So the idea is if we remove the fish, we can remove that nutrient source, and that could actually then lead to a lessening of red tide. If we could effectively starve it, at least of the fish source. So that's the premise of this study. Next slide, please. So we've conducted experiments at the Vester Field Station down in Bonita Beach. These are some of my graduate students here. Basically, we put dead mullet in these bins, let them cook in the sun, and we measure the nutrient releases. So it's a good practice on how to control gagging and smelling very wonderful smells from decaying fish. But we've gathered a lot of data from this. We've done this experiment twice, and we'll be gearing up to do it a third time later this spring to show reproducibility in the experiments. Next slide, please. So just to show you some of the data that's generated, we basically ran these studies for, I think it was 10 days, 2 weeks. And then you can see on the x-axis would be the time in hours. And then on the y-axis, the vertical axis, this is the phosphate release. And so the basically micromoles per gram wet weight of mullet per day. We tested 2 different temperatures. So you can see we tested 24, which would basically be an estuarine temperature. That's a lower salinity where red tide can actually multiply and grow and divide. It can survive below that, but it doesn't divide and continue to grow. And then 36 would be our representation of full strength salinity in the Gulf of Mexico. So what you can see here is basically after 96 hours, which would be 4 days, it tends to level off and then even drop off after 2 weeks. And we don't see much of a difference between the 2 salinities. Next slide, please. Most of the nitrogen was actually being released as ammonium, and we have a very similar pattern here where by 96 hours, it pretty much leveled off. You can see our error bars are fairly high, actually really high on a couple of cases here for the 24 salinity aspects, but there was no statistical difference between these. So again, this is from our first trial. We're waiting on the nutrient data from our second trial, and we'll see if we get similar results in our future studies. The importance of looking at the 2 salinities would be if you have different bacteria, different microbial flora, if there's an effect on salinity, on the nutrient release rates and how the nutrients are transformed, especially with the nitrogen species, or taken up by other organisms in the samples. We used ambient water, unfiltered water, so any critters potentially could have been in that water as well with this. Next slide. So this is basically a repeat of a study that was done in 2014 using pinfish. We did it on a much larger scale and for a longer period of time. Our phosphate release rates were lower than Kilberg Thornton, but our ammonium release rates were much higher. And so this is significant because this means that the decaying fish may be releasing more nitrogen and maybe a significant source of nitrogen for red tides. And Karenia brevis, red tide, prefers to take up ammonium. And so that is a very sensitive indicator, if you will, and factor, that driver, let's say, for potentially increasing red tides. And again, as I mentioned, we're awaiting the next nutrient analysis results. Next slide, please. So this is putting it in perspective. This was a nutrient budget put together by Cindy back when she was working with FWRI and some of the EcoHab work that was funded through NOAA. And so you can see here with the dead fish highlighted, if we're looking at 300% higher, all of these bars are going to be much higher. So basically what that means is dead fish could be a major player in terms of providing nitrogen to support red tides. Next slide. So what we're gearing up to do right now is we're basically waiting for a fish kill that's still basically offshore floating, not near the beach. It has to be offshore, that we can continue to sample around this and get field data to see if we see similar concentrations and release rates. So of course, as we're waiting, unfortunately, for a fish kill, we're not getting a good fish kill that's easily accessible and timely. You know, we can't go out there at 7 o'clock in the evening and try to sample it when we can't see it and things like that. So this is the next phase that we're looking at for this project, is to do some field validation. Next slide, please. We're also doing some composting. The idea is, what do you do with these dead fish? Do you just throw them in the landfill? Well, our idea is these are naturally occurring wild caught fish, if you will. They're organic, so you could actually compost fish and use it as an organic fertilizer. And there are several groups that are really interested in this. But the real question is, will the toxin degrade during composting? So we're doing these trial experiments with some of the dead mullet we collected in a smaller fish kill up in Midwest. And we're looking at these degradation rates, and we've been collecting samples. In fact, Tuesday we collected the set that will be doing toxin analyses to see if the toxins have in fact degraded. So this is a promising solution to these dead fish, but you know, we're still collecting the data to see if it's actually feasible. Next slide, please. So the next part of this study is basically an economic analysis. So if we remove these dead fish while they're still floating in the water, it's been estimated to be around $1,500 or so a ton, a ton of fish for removal. And so when you put that cost on a larger scale, you might be talking about millions of dollars. How much will red tide decrease with that? And what would be the economic benefit of that decrease? So we use the FWRI historical database, and we asked for data basically on a daily basis from all the sampling sites from Clearwater down to Marco Island. For each location, we put a 3-mile radius circle around it. Those are all the gray circles. And we have this run, we can automate this using GIS, and so we can see when red tide is present and at what times and where it is present. And the idea is we'll get the economic data before and after these different red tide events. And we can basically look at how tax receipts increase or decrease as red tide is present within these 3-mile radii. We're also collaborating with the University of Florida, who's doing a much larger scale version of this project. So we're trying to supplement their efforts with these efforts. Next slide, please. So basically, the first step in this, we looked at the tourist tax revenues because those were easy numbers to get access to. And so when red tide counts were greater than 100,000 cells per liter within any of those circles that I showed you, basically tourist tax revenues were 2.5% lower versus when red tide was at background levels. So this is for Lee County. So just in terms of the tourist tax revenues, that would equate to about $116,000 per month. So how does that then translate to other taxes and other lost business revenue, right? This is just looking at the tourist tax revenue. And so that's the next step in this, would be looking at how this translate into the larger scale. And then that's where this will dovetail nicely into the more in-depth project and analysis that the University of Florida is working on. Next slide, please. So lastly, I just want to finish up with looking at the resiliency of Brevitoxin in the environment. And so you can see the little thumbtacks on the map on the right here. There were 6 sites, actually 5 sites that we sampled. Estero Bay has its own thumbtack. I guess it's 6 sites. Site 1 and 2 are next to each other. That's what I thought. So 2 sites up in Sanibel and 4 sites in Estero Bay. We collected sediment cores, and then we basically sliced the sediment cores into 1 centimeter thick slices. And then we extracted them and ran them for toxin analyses up at Mote Marine Lab. And so you can see the toxin data on 3 of the sites on the graphs on the right. And so the main thing I want you to notice is that we are detecting toxin going down core. And this is a historical record. So this is going back in time. It's going back in time at least 50 years. So we're seeing that Brevitoxin can preserve in the sediments and represents a historical record going back decades. We need to refine this a bit. And this was actually an undergraduate project that you can see here with John Jarovsky. And he's continuing this work as part of his master. So we'll be refining this to see how far back in time we can detect Brevitoxin, and can we equate this to bloom events to see if blooms are becoming more frequent, more intense, so on and so forth. So just adding more data to try to address that question. Next slide, please. So that's basically a summary of some of the efforts that we're involved with here, just to kind of give you an update. And I don't know if there's any time for some questions. I have about 5 or 10 minutes here. Otherwise, thank you very much for your attention.
Dr. Parson, thank you so much for the information you presented. I know your availability might be the limiting factor in terms of times for questions. So I'm just going to open up the floor to anybody that has a question for Dr. Parsons.
Yes, sir. Mick Denham here. I'd like to ask a question, if I may.
Sure, please go ahead.
I thought that was a very excellent presentation. I thank you for that. You might want to think about trying to share this information as you presented to the mayors of coastal cities and also to the county. I think this information is the sort of information that the natural resource departments really would like to hear and help us explain this to our citizens and those people that do react to the red tide issue. So if you could find a way to share this, I think that would be excellent. That would be my question. Thank you.
Okay, great. Thank you very much. That's a great idea. And I am willing to do that. Yes.
Thank you.
Yeah, I have a question too. You're talking about like when the red tide outbreaks degrade, and you talked about them going into assistance and sedimenting out. Have you done any work into looking into like the viruses and maybe bacteria that and their role in ending outbreaks?
Collectively, the answer is yes. So for example, I have a graduate student who is looking at algicides. So those would be compounds that kill algae. That would be specific for red tide. And that student is up at Mote Marine Lab, and she's working with a couple researchers at Mote and up at University of Delaware. So I'm not directly involved in that project. I'm just advising the graduate student on that project. But there are several efforts looking at bacteriophages, viruses, I guess it would be algae phases, really, and some of these algicides. And Cindy Heil has a project that is focusing on the termination of red tides, specifically looking at these pathogens, not necessarily looking at the resting stage idea, because we don't have enough information to really get that funded yet. But looking at some of these viruses or these algicides that may kill the red tide. In terms of using it as a mitigation or control strategy, the problem with any of these strategies, including clay or ozonation or algicides, copper sulfate, regardless of the environmental issues, would be by the time we detect a bloom, it's too big and it covers too much area and there's no way we can address it. So we just need to understand its processes better. I mean, we can't even predict when a bloom's going to happen or when a bloom's going to go away. And so there's a lot of work that still needs to be done before we can look at how we can potentially mitigate and control the blooms.
You know, just a couple of follow-ups there, because I know that we had a volunteer who also volunteered for the for Crow on Sanibel, the Center for the Rehabilitation of Wildlife, and she was reporting turning in a lot of seabirds with that were verified to have red tide effects, well before any of the certainly the coastal readings showed any of this last red tide bloom. And the other thing is, you know, specifically viruses, you know, replicate themselves. And I thought I read somewhere that they play an important role in maintaining the diversity by keeping one species from being dominant.
Right. So yeah, it's a really good point about the cormorants. You know, basically the cormorants they told us first that red tide was here because they were very disoriented. I had, you know, people who were down on the beach send me videos of cormorants kind of stumbling around. And so brevitoxicosis is what we call that. And so, and talking with some of the fishing guides, they said they would notice like the thread herring and some of the other bait fish distressed and swimming up to the surface of the water and kind of swimming on their sides. And then cormorants or gulls or other birds would just be feasting on these bait fish that were basically suffering from red tide. And, you know, that may have been 10, 20, 30 miles offshore. We don't know exactly, but yes, definitely. Red tide was offshore before and affecting the birds before it came inshore. Actually, you're completely correct about the viruses, and there is a natural process with that, and they may control, you know, up to 99% of phytoplankton population dynamics. But when you see a bloom of red tide, the viruses are not necessarily controlling the population, or they're not controlling it to our liking. You know, maybe red tide would be much worse if there weren't viruses around. So there may be a lag effect on that as well. And a lot of my answer was focused on potentially using the viruses as a means of control. And that's where it gets a little bit more difficult. But altogether, I think we need to understand their role in red tide specifically.
Dr. Parsons, I would like to thank you very much for your presentation today. And Bill, I know you said you had a couple of questions. In the interest of time, I think we need to move along in the agenda, but I'd encourage you to contact Dr. Parsons directly, and anybody can with additional follow-up questions, if that's okay. Again, I appreciate the presentation. It's going to garner a lot of interest, and I assume we could spend hours with additional conversations. This was wonderful. Thank you for joining us today.
All right. Thank you very much, everybody.